Hello everyone, welcome back to another class on regeneration biology. In today's class, we will learn a new topic that is accessory limb model of regeneration, what we can learn from Axolotl and newt. We know when it comes to limb regeneration, the best studied, best explored model is Axolotl, and what we learn from them is having lot of depth and lots of similarity to that of zebrafish fin regeneration, but we have to accept the fact that fin is not a limb. Hence the research on limb regeneration is more on the axolotl. So this is a picture of axolotl. If you look carefully, you can notice something different. The hind limb is proper, but forelimb, you can see there is extra limb in this organism. That means we will see how are we bringing in extra limb while retaining one proper normal limb. The previous class also we discussed that if you have an injury and you express a cocktail of factors, you can get a accessory limb. So studies of ectopic limb formation tells us that all you need are specialized epidermis. That means specialized epidermis means an epidermis that is wounded, and a nerve connection, and fibroblast derived blastema cells. If you have these things, or if you take blastema cells from another individual and put it in an injury spot, you may end up getting an extra limb in that injury spot. So that is what people have been doing. This is mainly not to create a monstrous organism. That is not the goal. People want to understand how plastic the system is, how maneuverable the system is. So if a organism is capable of having multiple limbs that is originated, why can't mammal make at least the lost part? So this is what the researchers have been baffled, or researchers have been unable to come up with an answer that why a mammal do not regenerate. So let us see how to make an ectopic blastema. We have seen so far how blastema formed yesterday. In previous classes we have seen it. But now we are thinking about how an accessory limb or how an ectopic limb is created. First you need is to make a wound. Naturally wound signaling is must. Deviate a nerve to the wound site to stimulate the formation of an ectopic blastema. So what you understand from here, you can see in this picture, there are this is a limb, and this A stands for the anterior side, P stands for the posterior side. Anterior means this hand. It's like in my hand, this is the anterior side and the back is the posterior side. So that is what the concept you should have. When my limb is like this is anterior, that is facing front, and posterior facing back. So, or in other picture I can tell you, so in this is the anterior side and the back side is the posterior side. Here this is the anterior side, this is the posterior side. Okay, so if you make a injury and you take a nerve, that is the hand nerve, sciatic nerve, you deviate it and attach near the, or bring near the injured area, you end up getting a accessory blastema that is being formed on top of an existing limb. It's almost like I started getting a new limb formed from here. My limb has no issues, it is perfectly fine. So this is the picture of the ectopic blastema. So what has been done is, this is the nerve what you see inside, and it is cut and you made this nerve into near the wound site in a cartoonish manner. You can see it here. So how to make an ectopic arm? Graft a piece of skin, that is epidermis plus dermis, from the opposite side of the limb to the wound side. This is another approach what people have done. So this is the right and this is the left. And what people have done is, you can take a small tissue from the back, that is this side, posterior side, and put it into the anterior side of the same organism's other limb. So you took a tissue from here and put it in here, and you put a deviated nerve, and you end up getting, so now this has got an identity. It is my right hand, this is my left hand. So by putting a tissue graft from here and putting into the anterior side along with the injury and a nerve that has been put in, you end up getting my right hand that is being formed in my left hand, because this tissue also holds the identity. The injury formation, everything, injury is needed, deviation of the nerve is needed, but all they have done is you put a small tissue from the other side, and to the opposite arm, to the front side, you end up getting, as you can see here, you end up getting a this limb that is being formed on my left side as an accessory limb. So regeneration is a stepwise process, and this cartoon or this flowchart may look very complicated, but these are all congregating together. Signals can come from fibroblast, signals can come from the nerves, and also soon after injury, as you can see here, the blood clot is formed, and this is the timeline as you are seeing here. Regeneration is a very complex phenomenon, and it has got integrated sequence of events. Say you are sitting in one room, your brain knows I am sitting on the chair, I am sitting in a room, and that room is in so and so building, and that building is in so and so city. This much information your brain gathers. If this connection is gone, all of a sudden, if you lost that connection, you will not have an idea that where are you right now. You will not have an idea. While traveling also. That is why if you are traveling there are many people have done many research in that. Like you can see a lot of YouTube videos also. In that, people have gone inside one room, say for 72 hours. No clock, nothing. There is always persistent light. After some around 36, 40 hours, you completely lose your circadian rhythm. You have no idea what time it is. Is it morning, evening? You have no idea. Completely it gets reset. So while a injury occurred onto a limb, we are seeing just the injury, but lots of other things comes into picture. That is what has been shown in this flowchart. So this is a timeline up to 60 days, and blood clot is formed by 1 to 1 day. The blood clot is stably formed, and by around 5 to 7 day, wound closure and fibroblast migration, and around 10-15 days, myofibroblast and the collagen synthesis and wound contraction, that is the closing of the wound, and around 20 to 30 days scar forms. But while this is happening, you should understand there is a wound closure happening right from the injury site, and the proliferation and fibroblast migration happens. These are all influenced by various signals from the nerves, and also the signal is occurring from the blastema cells, and this will enhance the proliferation and dedifferentiation, and growth and pattern formation is important. This blastema, although we say it is a group of cells, they have to have identity, and accordingly you can see in a meagerly regenerating organism, scar forms in a no regeneration, whereas in regenerating organism, whether it is a skin or whether it has to form a proper limb, if it doesn't have signal coming from the nerves, then it will regenerate the skin. But if the signals are coming from the nerves also on that damaged area, you end up getting a proper limb or a potential accessory limb that can develop. So signals from the nerves are essential in an injury site of the skin, that the blastema formation is persistent, and the proliferation and ECM synthesis and chondrogenesis has to come down. And if it comes down, you will end up the so-called blast regress. So you don't want this ECM and chondrogenesis to come down. So how it is done? It is done by strong signals that is coming from fibroblast, which is also favored by the persistent nerve signaling. That is why nerve signaling can persistently occur in a wound site only by derooting a nerve. By attaching a nerve, you won't get that continuity. So then growth and the pattern formation happens, and you end up getting a proper limb regenerated. So you can see multiple stages are there. Scar forms in a non-regeneration, skin regeneration happens, and blast regression happens because it did not get the persistent signal in spite of having the nerve signal. But the persistent nerve signal allows the limb regeneration to happen properly. You can see here, understanding the pattern formation, that is basically the blueprint function of that, the orientation of the blastema cells. That means blastema is a mass of cells, but it has an identity which is anterior, which is posterior, which is dorsal, which is ventral, etc. So if you tweak it, you end up getting limbs like this, almost look like a branch of tree. You have got 3 limbs coming from one stack. So organ, despite having lot of emergent properties, the blueprint can be distorted by tweaking the orientation of the blastema cells. Or like I told you, take skin from here and put in here after cutting. So this also can have lots of implication. You can also see the connective tissue fibroblast are the progenitors of early blastema cells. That means the fibroblasts have the positional information. Like, although in this picture you are seeing fibroblast, but this fibroblast knows where it is belonging to, and this fibroblast know where it is belonging to, and this fibroblast also knows where it is belonging to, and it will act accordingly. So positional interactions between fibroblasts also plays a major role. Although fibroblasts are present, blastema cells are also present, and each of these cells, the congregation of both this mesenchyme cells, which has the potential of making an restoring a damaged part, and also the fibroblast cell, and there is an interaction between them also. And so the positional interaction between fibroblasts create the blueprint of limb regeneration. If you distort it, as you can see in this picture, say this is a blastema, anterior, posterior. Anterior means this, posterior means this. Anterior, posterior. And the newly formed blastema, you twist it. That means if this is anterior, this is posterior, and the blastema formed, you twisted this way. This is cut it and you twist it. As you can see here, anterior, posterior, and then posterior, anterior, you fused it. As you can see here, then you end up getting completely different limb. Instead of a limb formed like this, it will now form like this. So that is what you can see in this picture, anterior, posterior, and now anterior, posterior. You end up getting a multi-fingered limb. And people have done lots of such, mix and match combination. The outcome of regeneration is dependent on the positional information. Now you can see here, anterior, anterior, posterior, anterior, 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 posterior, anterior. This is the scenario. But what they have done is anterior, anterior, you put with anterior, anterior. So what they have done is taken the posterior and removed and replaced with an anterior. So whatever they have, the entire blastema has got only anterior information. And then on posterior, anterior, you are replacing with the posterior, anterior. That gives a normal limb, because posterior, anterior is now restored, fused with another posterior, anterior. Here another anterior, anterior they did with anterior, posterior. Then you end up getting a multi-limbed structure. And posterior, anterior didn't fused with the anterior, posterior. Then you end up getting a multi-limbed structure. Here only anterior part is kept, and it has got only one toe, that is somewhat equal to your thumb. That only is being formed. And this one, posterior, anterior, is fused with the posterior, anterior. Same thing would have happened anterior, posterior fused with another anterior, posterior. You will still get a normal limb, even though the blastema is from another source. So the growth and pattern formations are controlled by the local cell interaction. So this is the proof that this is a blastema, and how you handle the blastema. Blastema's positional information, you can tweak it across different positions within that blastema. You end up getting this kind of cells, this kind of limb structure. The creation of the amphibian regeneration blastema may also depend on maintaining ion currents through the stump. Stump means that growth stump. If this electric field is suppressed, the regeneration blastema fails to form. So we should understand the electric current. It has got a strong influence by neurons and also by the innate properties of the cells, because we know interior of the cell membrane is negatively charged and outside is positively charged. Skin flaps inhibit both the current of injury at the amputation surface and regeneration of that limb shown in newts. This is a published paper. Such fields have shown that it is important for the regeneration of tails of Xenopus levis. So the same orientation identity is followed in the tail regeneration as well. In this frog, where VATPase, that is a gene, that is the proton pump, gets activated within 6 hours after tail amputation, changing the membrane potential and establishing the flow of protons through the blastema is the role of this VATPs. If this proton pump is inactivated, there are different ways in which you can inactivate, either by mutation or by the drugs. The depolarization of the blastema cells fails to occur and there is no regeneration. So depolarization means the interior membranes inside is negatively charged, it becomes positively charged locally. That is depolarization. That is the principle lying behind every neurotransmission. Every, like you are seeing something, an action potential is created because of the depolarization. So if this proton pump is inactivated, then it can be done by various methods and VATPs plays a major role for that. You will not get a regeneration response at all. So this has also been done in published in a paper, that is H plus pump dependent change in the membrane voltage are an early mechanism necessary and sufficient to induce xenopus tail regeneration. Those who are interested can read that article. So bone, dermis and cartilage just beneath the site of amputation contribute to the regeneration blastema, as do the satellite cells from the nearby muscles. So they also, bone, dermis also plays a role. Unlike the flatworm, where the regeneration blastema tissue comes from the blueprint of stem cells, that is ceneoblast, we have seen it in the previous class. In the salamander, most of the regeneration blastema appears to arise from dedifferentiation of adult cells, followed by cell division and redifferentiation of those cells back into their original cell types. So this is how normally happens, normal regeneration happens in an amputated organism. However, there is lot of controversy about whether this is true dedifferentiation of normally post-mitotic cells, or whether much of the limb blastema is formed from the activation of uncommitted stem cells residing within the adult tissue. Means whether they are present in every tissue, like skin has got skin stem cells, your retina has got rod progenitor cells, whether those kind of cells are coming and expanding in number, or they are formed newly, de novo formation. So many such debates also going on, but it is well accepted that it most likely the existing tissue type dedifferentiate. But do we have some evidence? Can we show it? So a major question is, do the blastema cells keep a memory of what they have been? We say blastema is formed by the dedifferentiation, reprogramming of the cells present in that injury site. But if that case, do those cells carry some memory? Do we have some proof? Kragel and colleagues in 2009 found that the blastema is not simply a collection of homogeneous, fully dedifferentiated cells. Rather, in the regenerating limb of salamander muscle cells arise from the previous muscle cells. So previous muscle cells reprogram, become blastema, then give rise to the new muscle cells. Dermal cells form from only the old dermal cells, and the cartilage cells can arise from only pre-existing old cartilage cells or old dermal cells. Thus, the blastema is not simply a collection of unspecified multipotent progenitor cells. It's like if you have in the limb, you have got muscle, nerve, skin, blood, bone, cartilage. All those stem cells are derived from those tissues. No tissue is going back. Bone is making its own contribution, muscle is making its own contribution, skin is making its own contribution. Now they all look like a undifferentiated or dedifferentiated stem cell like, but they have still the identity from where I have come. Just like if you go to America or London, you still know what is your village name, from where you belong to. Even you may tell to your kids, oh, this is our hometown. So such an identity is there. So thus the blastema is not simply a connection of unspecified cells. Rather, the cells retain their specification, and the blastema is a heterogeneous assortment of restricted progenitor cells. They are all progenitors, but they have an identity. So blastema cells retain their specification even though they are capable of differentiation. So What is done is they have a cartilage, a GFP expressing limb, and they took a bone cartilage or a cartilage tissue and transplanted into a wild type limb. So GFP expressing one small limb is taken and put it into a wild type limb of a recipient, and then they cut it. Here they created a cut, and then they saw that these GFP are the newly formed blastoma cells have the GFP, and those GFP bearing blastoma cells are giving rise to only the cartilage. See, you can see the grafting is been done and the amputation is done right in the place of graft, so that the GFP transplant or the transplanted GFP bearing cartilage is injured, and now the blastoma is formed from there, and that GFP is now getting into only bone. It doesn't give muscle, it doesn't give rise to skin, it gave rise to regeneration. The newly formed limb bone cartilage had this GFP. So cartilage from the specific portion of the limb of salamander expressing GFP is transplanted into an equivalent position in the cartilage of salamander limb that do not express GFP. So the grafted tissue integrates within the host tissue. Naturally, salamander or axolotl do not throw or reject tissues. Amputation is performed in the middle of the grafted tissue. That means it has GFP. That grafted tissue is injured. The blastoma forms containing GFP expressing cells. The regenerated limb is studied to see if the GFP negative or GFP expressing cells are present only in the regenerated cartilage or other tissues. What they found, it is present only in the regenerated cartilage tissue, not in other tissues. So this is another article published in Nature. Cells keep memory of their tissue origin during axolotl limb regeneration. Those who are interested can read this article, because that is one good discovery that happened, that it retained, the blastoma retained the memory of from where they originated from. Longitudinal section of the regenerating limb 30 days after amputation. Muscle cells are stained in red, nuclei are stained in blue. The majority of the GFP positive cells shown in green color are seen in the regenerated cartilage only, because cartilage only had the GFP from the donor, and not in the other regenerated tissues such as muscle. So the proliferation of blastoma cells, that is the requirement of the nerves and apical ectodermal cells. So we have seen in the beginning of the class that the nerves are important. Let us see how nerves can contribute. So the AEC, apical ectodermal cap, stimulates the growth of the blastoma by secreting a factor called FGF8, just like the AER, apical ectodermal ridge, that does during the normal limb development. But the effect of the AEC is only possible when the nerves are present. Even though you have an apical ectodermal cap is present, if nerve is not there, you will not get the regeneration. Singer in 1954 demonstrated that a minimum number of nerve fibers must be present for the regeneration to take place. Remember the year? It is done in 1954. The neurons are believed to secrete factors necessary for the proliferation of the blastoma cells. Among the many candidates for nerve-derived blastoma mitogens, the best example known, that is the newt anterior gradient protein. It is also known as NAG, newt anterior gradient protein, secreted from the Schwann cells. Remember, not from the neuron, it is secreted from the Schwann cells, which is the glial, supporting glial cells of the peripheral nervous system. Your limbs have the peripheral nervous system. Only brain and spinal cord is central nervous system. So the Schwann cells secrete this newt anterior gradient protein known as NAG. This protein permits normal regeneration in limbs that have been denervated. So if nerve is not there, amputation happened, but if you put NAG in that spot, then the regeneration can happen. Nerve is not there. If nerve is there, naturally the Schwann cells will be there. If nerve is not there, Schwann cells will not be there. Hence NAG will not be there. But if you supply NAG, you can end up getting the limb regeneration. So this protein permits normal regeneration in limbs that have been denervated. That means the nerve has been removed. Axolotl use something called a neuregulin secretion from the neuron, that is of the sciatic nerve. It is not produced by the Schwann cells, it is produced by the sciatic nerve itself as a major factor contributing to the regeneration of the limb. You can see here the right limb denervated. You can see here in this picture, it is denervation happened, and then so right limb is denervated and both limbs are amputated and electroporation with NAG. So what you can see here, the nerve is removed and it's amputated, but nerve is not there. Whereas in left, there is amputation, but nerve also is there. And now this is not supposed to regenerate. But if you put NAG, then like this limb, this also will regenerate despite not having the nerve. So you can read about the molecular basis of nerve-dependent limb regeneration in adult vertebrates, an article published in Science, very interesting article. Those who are interested in this field can read about it. So here this is a picture from article published in Developmental Biology, and also this has been discussed well in Current Opinion in Genetics and Development in 2008. So this is a newt limb, and which is normally present, and it has been cut. But in here what has happened is they have removed the sciatic nerve. You can see the wound healed after cutting. Left limb is intact, and left limb, even if you cut it, it can regenerate. Since new nerve is removed right from here where the star mark is there, it is removed, the wound heals. It doesn't give rise to. But if you give NAG, you administer the NAG by electroporation, you end up getting the limb that is being formed. So these are all indicators of new regulators of vertebrate appendage regeneration. But you may have remember, despite all the odds, here this complex picture which we discussed, we should understand that each of them, you cannot assume that NAG is good enough or neuregulin is good enough. But the other signaling, like the blastema causes its own signaling, or the AEC, apical ectodermal cap, creates the FGF8 signaling, and also the involvement of delta notch signaling or Wnt signaling. These are all cannot be sidelined because they are important. They do contribute. Otherwise, what will happen? You end up getting scar that will be formed. Like if you can see here, in each of this regeneration approach, there will be formation of this scar in the first place. Later, this scar has to be dissolved, and this happens because of the proper induction of lot of metalloproteases and collagen degrading enzymes, so that the scar will be removed and the cells will be replaced, and these cells now differentiate and form the proper limb structure. So we should understand, in the same animal, despite having nerve properly present in this limb, even if you cut it, that nerve is not coming to help for the adjacent limb, because if there is an endocrine factor, it is secreted into the bloodstream. Naturally, whatever receptors that is making use of NAG or neuregulin must be present in here also. So they are not available through the bloodstream. If they were available through the bloodstream, it would have been able to cause a proper regenerative response. So just like in mammals, after injury, there is a wound healing response, and also there is a scar formed. But the scar gets dissolved because the early deposition of the blood clot, which has got fibronectin, etc., allows the migration of the cells into that site, and they can now also retain the positional identity, so that the limb is formed forward, not formed backward. But you can make it form backward if you twist the anterior into the posterior side and posterior into the anterior side. You can end up getting a limb grown this direction. So that much plastic is this system. So we will learn more about the nerve dependent limb regeneration in the subsequent classes. Thank you.